All right, ready to start sharing?
Yes.
All right. All right. Yeah, so my name is Michaela Coates. I'm also here with my teammates, Kai Dang and Carmen Gardner, but Carmen's actually going to kick us off today.
So we're here to talk to you all about abalone. To tell you a little about who we are, we recently graduated from University of California Irvine's Master of Conservation and Restoration Science program. The program is a professional degree program designed to provide graduates with the skills and knowledge necessary to hold leadership and management positions in conservation, restoration, and sustainability fields. Our community partner for our capstone research project was the National Oceanic and Atmospheric Administration. Specifically, we work towards, oh, I think I was muted.
You're good now.
Sorry, okay. So to tell you a little about who we are, we've recently graduated from University of California Irvine's Master of Conservation and Restoration Science program. The program is a professional degree program designed to provide graduates that with the skills and knowledge necessary to hold leadership and management positions in conservation and restoration fields. Our community partner for our capstone project was National Oceanic and Atmospheric Administration. Specifically, we work towards Black abalone conservation goals designed by the National Marine Fisheries Service, informally known as NOAA Fisheries. Our discussion will start with addressing what abalone are and more about their biology and natural history. We will then go into species management of the different kinds of abalone. We will focus more specifically on Black abalone listing and recovery and our capstone project, Black abalone habitat assessment. And then we'll finish up with future directions of how our project can be utilized towards abalone restoration goals. So what are abalone? Abalone are a marine gastropod species commonly found throughout California's rocky intertidal zone. They are large marine mollusks and their nickname is earshell from the shape of the spiral. They have respiratory holes for breathing and breeding. They have a muscular foot, epipodium, and tentacles as shown on the bottom picture. The blood or hemolymph of abalone has no clotting factors. So that means cuts, punctures, or lacerations can cause them to bleed out and die. Abalone are broadcast spawners, which means they require close proximity between males and females for successful restoration or fertilization. Abalone have small home ranges. A study done on pink abalone in 2014 found their home ranges to be less than 183 meters and greater than 2 meters. So they mostly only move back and forth between a few locations. At their home location, they often hollow out the rock a bit around themselves, forming a home scar that they fit into perfectly, shown in the bottom right picture. Models have shown that this homing behavior enables them to reproduce more successfully. So what do abalone eat? They eat algae and kelp, which commonly drift through to the sub and intertidal zones from offshore kelp beds. Juveniles and adults have shown preferences toward giant kelp and feather boa kelp, which are both common throughout the species range, but exhibit annual variability. Adults catch kelp by reaching out and grabbing kelp drifting along the seabed with their muscular foot. Abalone played a key role in Native American culture. Indigenous would eat the meat and use the shells as bowls. They would utilize pieces of the shell to make fish hooks, scrapers, beads, necklaces, and decorations, and even bartered with the shells. Historically, hard hat diving came about along the West Coast in the 1890s. In 1920, divers were said to have gathered about 15 dozen abalone per day. By 1930, divers would gather about 30 to 60 dozen a day. And in the 1920s in San Diego, skin diving became the new sport after shallow need to hip deep waters were cleaned out by shore pickers. In 1933, the Bottom Scratchers of La Jolla became the world's first dive club and became influential after requiring prospective members to dive to 30 feet, capture 3 abalone on a single breath, and bring up a good-sized lobster and grab a 5-foot horned shark by its tail. The bottom picture shows a pry bar, a tool used to pluck abalone off of their cracks and crevices.
So not only has recreational fisheries been a prominent culture within the West Coast of the United States, but there was also very, very large commercial fisheries for many abalone species in this region. So as of that, they were subjected to very high rates of overfishing. I think this photo is so extremely powerful in demonstrating the absolute massive quantities in which abalone were harvested. And as such, a combination of overfishing plus other factors such as disease, as well as natural predation, led to massive declines in populations across abalone species in the past about half century, and one after another, the commercial and recreational fisheries started closing. About 7 species can be found in California, 7 now that pinto and threaded are considered to be one species, with variants in the northern and the southern range. That includes pinto, flat, black, red, pink, green, and white. This dark blue box here that I created is Southern California. So, we're up in Orange County, but you guys are all down in San Diego. And so as you can see, this region is within the range of all of these different species. Since they started to see these massive declines and all of the recreational and the commercial fisheries started to close, the California Department of Fish and Wildlife developed the Abalone Recovery and Management Plan, which was published in 2005. This includes protections and regulations and management structures for all of these different species that you can find within California. This includes interim plans for recovery that, of course, first means reversing the declines and the rates of population decline, particularly for those species that are approaching extinction, and as well for management is managing the red abalone recreational fishery, which I will talk more about later when I dive more into that species a little bit. For their long-term plans for recovery, it's to rebuild populations in 3/4 of the historic range. When it comes to these recovery plans, they tend to have very specific quantitative metrics in order to identify the success of recovery. And then from a management perspective, in the long term, they hope to develop zonal management of these fisheries. Hopefully they can open once again with successful recovery. Like I mentioned, at this point, all commercial and recreational fisheries are closed except for the recreational fishery for red abalone in Northern California, and 2 species, the white and the black, each have their own recovery plans due to their listing on the Endangered Species Act. And so for those reasons, these 3 are kind of outliers in the sense that they have special cases. So I will be talking a little bit more about these 3 species more in detail. So red abalone is pictured here, one of the largest abalone species, growing up to be about 12 inches long. Its size has helped make it a very important fisheries species. You can imagine it having more meat as it gets bigger. Like I said, the recreational fishery of red abalone is still allowed only north of San Francisco Bay, and there are still a ton of regulations, including the prohibition of scuba diving. So it is only free diving or picking off the rocks. However, since they can commonly be found up to 130 feet deep, anything past the capacity of free diving acts like a little sanctuary for the species. However, they can be found more intertidally, again, in the northern portions of the range, but they are a cold water species, so they will remain in deeper waters or near upwelling zones in more southern regions. But again, for the recreational fishery, everything from season to size to the tools used to the number harvested each day is all regulated. However, due to population die-offs that they believe is related to kelp forest collapse, the recreational fishery is to remain closed until 2026. So for the next 4 years, and then I believe they will be reevaluating the population status. The up and coming alternative, however, to fishing wild populations is aquacultured abalone. From what I've seen, at least in the aquaculture farms in California, red abalone is the species that is typically reared and then sold to markets and restaurants. Pictured here is the cultured abalone farm in Goleta, California, which our advisor luckily got to visit with some students a few months back. We weren't included, but maybe one day I'll get the chance to go see it. And a fun fact that I learned is the owner of this farm is also a member of the NOAA Fisheries White Abalone Recovery Team, which is the species that we will be talking about next. So white abalone are one of the deepest living abalone species, going to about 180 feet deep. They can grow to be about 10 inches long. And this species is particularly special because it became the very first marine invertebrate to be listed on the Endangered Species Act. And now that's not necessarily a great thing to be #1 at, but the story of their recovery is one that will hopefully, and has already been, having impacts beyond just this one individual species. So their fishery closed in 1997, and that started to slow the decline. But as Carmen said, they are broadcast spawners. And so low densities led to very low recovery. They were listed as endangered on the Endangered Species Act in 2001. And at the time of their listing, the National Marine Fisheries Service actually did not designate critical habitat out of fear that the public learning about locations where there is good habitat might increase rates of poaching. And so then later, 7 years, the recovery plan was published in 2008. The recovery plan for white abalone focused on a few major recovery actions. The first few, of course, were to monitor and protect the populations existing in the wild as well as their habitat. But of course, again, low densities meant very low reproductive and recovery rates for abalone. So they decided to also employ captive breeding programs to enhance wild populations. Here, this photo on the right is a diver collecting a white abalone in 2016. And the purpose of doing so was to inject the new genes from the wild stock into the captive broodstock population that will eventually be outplanted. And captive breeding abalone is not as easy as it might sound. It requires mimicking the environmental and the chemical cues that trigger spawning in the species. However, as of 2019, or in 2019, 10 white abalone adults produced and fertilized 25 million eggs. So there is a great progress being made, and luckily enough, this provided a framework for the next abalone species to become listed, which was the black abalone, and the kind of main character of the research that we will be talking about for the remainder of the presentation. So black abalone can grow to be about 8 inches long, and this is the species that will inhabit the middle to the lower intertidal zones. They can be found all the way up into the upper middle zones, and their shell is this beautiful bluish black color that you see here. They currently range from Point Arenas, California to Bahia Tortugas, Mexico, which can be seen in green. However, the extent of their range contraction is the red portions on this map, and that red box in the middle is the study area for our research of Orange County, California. Now, as you can see on the photo of the left, black abalone used to be extremely abundant. This is a photo taken in the intertidal zone of Laguna Beach in 1930s, and you can see it seems to be quite difficult to walk across the rocks without potentially stepping on a black abalone. But of course, that is no longer the case now due to a combination of overfishing and, most significantly, the spread of a disease called withering syndrome. This disease was first observed in the 1980s, and it affects the species' digestive organs so that it can't produce digestive enzymes. And it also atrophies their muscular foot, which is seen on the photo on the top right. Those two kind of combination of factors can lead to mortality through starvation and or predation. And so as such, they experience declines in population of up to 99% throughout portions of their range. And most notably, the southern portions of their range, as withering syndrome has been seen to be exacerbated with warmer sea surface temperatures. Now, with the loss of a species, of course, also comes the loss of their ecological role. And as an herbivorous consumer, abalone have been seen to influence the community structure through the maintenance of substrata. Miner et al. 2006 found that decreases in abalone abundance have been correlated with decreases in percent cover of crustose coralline algae and bare rock, and increases in percent cover of invertebrates, which not only creates a barrier to their own recovery, but limits recruitment for other intertidal species as well. And of course, as Carmen already mentioned, they are an important cultural and financial resource for the indigenous communities of the West Coast, diving communities, and of course, the economic value of their fishery, which closed in 1993. So the rest of their timeline, fishery closed in 1993. They were formally listed on the Endangered Species Act in 2009. Their critical habitat was designated 2 years following. And then just last year, their final recovery plan was published. This recovery plan was very similar to that of the white abalone. It outlined 8 major recovery actions that focus on supporting natural recovery and enhancing local populations through methods such as captive breeding and outplanting, and protecting and restoring black abalone habitat. This photo here is black abalone spawning holding chambers at the Southwest Fisheries Science Lab. And it was seen that black abalone were not faring as well with the captive breeding process. They are found to be a little bit more particular and sensitive when it came to their spawning conditions. But as of 2019, they did have a breakthrough of their first induced spawning. So again, great progress is being made in this portion of their recovery plan. And now, when critical habitat was being designated, it existed in segments throughout their range, including offshore islands such as the Channel Islands. However, the Orange County coastline was excluded in part due to their local extinction. However, population surveys done in the past few years have found a handful of individuals scattered across the region. And the combination of historical occupation and the potential success of restoration and transplantation lead us to believe that this region can support black abalone recovery. However, local habitat surveys in this region, or recent surveys in this region, have focused solely on geomorphology. And so the knowledge gap that our research was aiming to fill, or I suppose it passed, so hopefully did fill, is a more comprehensive understanding of habitat suitability of rocky intertidal sites, as well as population status in Orange County, California.
So we addressed 3 research questions. 1, we answered which intertidal sites along the Orange County coast provide suitable habitat for black abalone through habitat surveys. Then we answered which sites have highest rates of human visitation and impactful behaviors using MPA watch data. Finally, we found which sites have the most abundant and persistent offshore kelp biomass using remote sensing data. So we assessed several habitat factors in this study. We assessed geomorphology, which is the complexity of the rock structure at each site, and assessed the total surface area and quality of available habitat. We assessed biotic interactions, and which include interspecific interactions, food availability, and predators. And lastly, we assessed human visitation to habitat and human behaviors at each site. In addition to food availability within segments, we looked at food availability offshore, specifically by using giant kelp remote sensing data. This is an important factor because looking at black abalone recruitment success, because healthy kelp forests offshore are critical to providing consistent food resources. We first followed UC Santa Cruz's established black abalone protocol, which is used across the state to assess geomorphology. Last year, MCRS students built on this protocol by including belt transects. We estimated the percent of good, moderate, and poor habitat shown in the picture to the left. We assessed substrate relief, sediment, and exposure. We used a random number generator to get 10 points along a 50-meter transect to measure the area of cracks and crevices and area of boulders within perpendicular 5-meter transects of each random point. For biotic interactions within segments, we estimated the percent cover of biotic organisms in the 5 best cracks and crevices surveyed. We completed 30-minute time searches at each segment for predators and measured sea stars from the center of their disc to the tip of their longest ray with a caliper. We surveyed the entire segment for presence or absence of macroalgae and noted it was in situ or drift. To further assess food availability, we analyzed abundance, variability, and long-term persistence of giant kelp biomass remote sensing data from 1984 to 2020 provided by Dr. Tom Bell of UCSB. We created 10 2x2 kilometer boundaries to extract the time series data from these specific locations shown on the map in orange. We assess abundance by looking at the current and historical means. Variability was assessed across sites by analyzing coefficient of variation and consecutive disparity. And long-term persistence was analyzed by looking at the number of years with zero kelp. To assess human impacts, we looked at human visitation to the specific abalone habitat as the number of people per site in the area during a 2-hour survey. The sites along the Orange County coastline range from south of Newport Harbor down to Dana Point Harbor. We recorded MPA status as our sites were located within 3 different MPAs shown in the map. We then analyzed citizen science data collected on human interactions during a walking survey that was provided as MPA watch data from the OC Coastkeeper. So here are 2 maps of our 18 habitat assessment segments within sites and are shown with an aerial imagery background. If you refer to Gough Island and Treasure Island in the bottom left corner, the aqua colored polygons are habitat segments where we completed our surveys, and the light orange buffer shows the approximate range we created to assess human visitation.
So this is our scoring system. So after we survey 10 categories for each site, then we give them a score. Either it can be positive, neutral, or negative. And on the tables, you will see the white rows were scores assigned by categorical criteria, and the blue rows are score assigned by quartile values. So those quartile values are actually just relative to each other. So we gathered the data for 18 sites. We have a list of those data, and then we calculate the quartile values. So any values that is lower than quartile 1 mean they belong to the lower 25% of the data. Those higher than quartile 3 mean they belong to the upper 25%. And then in between quartile 1 and 3 are those that are the rest 50% of the data. And so before we go deeper into the results, I just want to let you know that I will briefly go over all of the categories that we mentioned, but we want to focus more on the human impact and offshore kelp, because those are additional aspects that we added to this year project. And these 2 are quite important to black abalone as well. So this is the map of MPA violations and enforcement presence that we have in our site. So on the left side of the graph, you will see these are the sites that we put in a geographical order from north to south. And we also group them in MPA regulations. So we have Crystal Cove SMCA, we have Laguna Beach SMR, and we have Dana Point SMCA. The red line on the red bar is the MPA violations, and the green bar is the enforcement present rate during each survey. So we make some comparison between different type of MPA, and we found that there is no significant difference between MPA enforcement presence between SMCA and SMR. So even though SMR is more restricted, SMCA and SMR has the same enforcement presence. However, SMCA actually has a significantly higher violation rate than SMR. And we found that there is a very weak correlation between enforcement presence and APA violations. So the takeaway is that just even if we increase APA enforcement presence, there's no guarantees that we will lower APA violations. So there have to be something else that we need to do to reduce the violations, not just like having more rangers, even though like they are doing great job at it, but they cannot be there all the time. And this is the data of our kelp remote sensing. So because kelp actually die back every year, and then they die and then they grow back. So if we just use quarterly data, it will be really messy and we won't be able to get some informative information. So this is actually a moving average of the kelp biomass. And what it means is we take the average every 5 years. So we have the average of biomass from year 1 to 5, then from year 2 to 6, and then 3 to 7, and so on. And to have this graph. And for this graph, we're gonna look at kelp stability in each site. And we break it down into 3 smaller categories. We have the average biomass, which is kind of like the line itself. So if you look at the line, you want to see higher peak. That just means that we have higher biomass, and that is the better. And then you want something to be more stable, so it is less varied. So you want the graphs to be less like fluctuating. And the last thing is you want them to last a lot longer and be more consistent. So if you look at the red box right here, you will see that this is a very long period of very, very low kelp biomass, and abalone don't really like that. So we don't want that to happen as well. And on this graph, we also mark some of the big El Nino years, and you can see how that El Nino years can affect kelp biomass. Like you will see significant reduce of kelp biomass after El Nino years. And because of that, we look at the last 5 years after the final big El Ninos, just so we can see like how's the Kelvin's doing, and we can maybe make some prediction in the future of where should we outplant our abalone if we have some success in that. And then we also mark the year from about 2002 to 2012. Those are the years that Nancy Caruso started doing her kelp restoration project. And so this is the numbers, the statistical numbers that we have from the graphs. So the first 2 columns are abundance. You want these numbers to be high because that just means we have more kelp. And then column 3 and 4 for variations. So you want the least variation as possible. the last two is for persistence. So these are like how many years, how many consecutive years that we have very low number of kelp. So you want these numbers to be small as well. And the sites, we also put these sites in geographic order from north to south. And if you look at it, we color coded these so that we have red is not so good, and then we have green is doing a lot better. So we have kelp from the south of Orange County is doing better than kelp from the north of Orange County. And well, this might look like a complicated table, but and I will walk through it with you guys, but the takeaway is we did a thorough job with this. Yeah, so if you look, when you look at the first 3 rows, these are the protocols that we use to assess the geomorphological of the area. So we look at rocks, cracks, and crevices, and we found that they are quite consistent. So there might be a few places that they have negative sign, but overall, when you combine all these 3, Orange County is quite good for abalone in terms of cracks and crevices, which is expected because historically there were a lot of abalone in this area. And then we look at the intertidal kelp and offshore kelp because they are the food source, and we actually found a lot of kelp wash up into the intertidal zone. So in terms of intertidal kelp, they are doing quite well. And because of that, we want to look at offshore kelp as well, because more offshore kelp means more wash up intertidal kelp. And like I mentioned before, south of Orange County is doing better in terms of offshore kelp than north of Orange County. And then for interspecific interactions and predators, these are pretty much like kind of like the neighbors of abalone. And we would say that Orange County has a very welcoming neighborhood for abalone. And then we try to make it like a positive thing when we include human presence as well, but we find there were a lot of violations because we have a lot of human visits, and then we have like a lot of violation in terms of human behaviors, but these can be mitigated. And hopefully with all of your help, we can make it a lot better for abalone. MPA status, well, we cannot change these. It's not easy to change. And I just want to mention again that just because some of these are negative, it doesn't mean that they are really bad for abalone. Sometimes it is just like for MPA status, negative area may just mean they are the least restricted area. It's not that like they are bad or anything. It's just least restricted.
So as you could see from that last slide, this habitat assessment that we did was extremely complicated. We had to take into account quantitative data as well as qualitative assessments. And like Kai mentioned before, we wanted to think about if a factor ranked negatively or maybe even neutral, could that factor be potentially mitigated through things like enforcement and education or habitat restoration? And so these are our final recommendations. Unless you guys happen to be familiar with the Orange County coastline, these sites might not mean as much to you, but in the end, we recommended to prioritize Shaw's Cove Segment 2, Gough Island Segment 1, Dana Point 2, and Moss Cove 2 for restoration of black abalone, as they all contained complex geomorphology, had high rates of positive biotic interactions, and we realized the human impacts at those sites could be mitigated. However, oppositely, we do not prioritize Crystal Cove State Park 2, Woods Cove Segment 2, or either of the Little Corona Del Mar sites, as they all had poor geomorphology, as well as poor biotic interactions that, especially when coupled with weak offshore kelp availability, and extremely high rates of human impacts, including direct MPA violations, it would be relatively inefficient to restore, especially when comparing to the sites that did better in our final assessments. So this assessment that we did, like we mentioned earlier, continued to develop on the protocol that was developed by the team that came before us, which developed upon UC Santa Cruz's Black Abalone protocol. While it did take in a little bit more effort, it took our team of 3 about 2 to 3 hours to complete the fieldwork component of this study for each site. It did provide us with a more comprehensive and quantitative approach to measuring habitat quality. When we compared our habitat assessment results to the results of that of the UCSC protocol, there was some overlap, but our results did indicate that some sites were better or worse than the UC Santa Cruz protocol did, since we looked at the habitat factors that abalone require at a more detailed scale. And so this new assessment protocol that we helped develop could be used to assess other sites or regions for the potential recovery of black abalone, and our results would be used to inform the future recovery of black abalone in Orange County, California. Thank you guys all so much for listening. We're so glad that we were able to come and speak with you guys today and share our research and share our love for abalone with a wider audience.
Yeah, thank you. I know that we have a few questions here, at least a couple to start out in the chat. And then if anyone else had any further questions for Kai, Carmen, or Michaela, please go ahead and put them in the chat so we can get to those for you. One, I think, was back when you were presenting, Michaela, if an abalone was removed from their home for whatever reason, what's the best way to replace the abalone safely?
For the sake of clarifying, is this asking about whether an abalone was like dislodged from the substrate, or was it more so in a area that you might not think is best for the abalone?
This was from Jackie or Warren. So if you guys want to go ahead and unmute, you can clarify that for her.
It's removed from the substrate.
If it was dislodged from the substrate, I suppose, trying to move them deeper into the intertidal and maybe guarding them, watching them to make sure that they do reattach to the substrate safely. I did have a conversation with, again, Nancy Caruso. She runs a nonprofit called Get Inspired, which has been doing a lot of kelp as well as green abalone restoration. And she was warning me that abalone, which I believe down at you guys would be mostly green abalones, a disease has also been spreading through both wild and captive populations. And so there would be a chance that some abalone have been washing up onto shore unattached to substrate. And so I think the same thing would happen for that. But again, it's also recommended that if an abalone just maybe looks like it's not in the place it's supposed to, but it is healthily and happily attached to a substrate, to of course never bother it or move it.
All right, did that help, Warren?
Yes, thanks.
Wonderful. The other question you had then, this is also from Warren. Do we know the causes of the low kelp biomass years?
So most often the giant declines that we saw in that moving average graph happen to do with El Nino. Kelp forests do not fare well with higher sea surface temperatures, and it brings about a lot of storms that will also kind of break those kelp forests off, and that leads to them being washed up in the intertidal. But it exhibits a lot of variability annually as well as cyclically with different climate cycles that happen.
All right, and we have another question from Scott here. Is the ultimate goal for the sites you identified to populate with captive bred abalone? How successful have other reintroduction programs been?
So it could be, yes. So there's a combination of, captive bred abalone that are outplanted. Another option would be to transplant wild abalone from, there's healthier populations such as out in the Channel Islands. So both transplantation of captive bred stock as well as the transplantation of wild stock. And in terms of the success of reintroduction, I believe they had introduced some white abalone recently. Amy, if you know anything more about that, you can feel free to chime in. But they are still, of course, very secretive about it. Since it is an endangered species, they are not going to be telling people where they are doing things like that.
Yeah, hi folks, I'm Amy Henry. I'm an instructor at UC Irvine and this team's advisor from their master's program. And abalone restoration and reintroduction programs have been implemented in a couple places. They've been mildly successful in the Seattle area with pinto abalone. Green abalone restoration in Orange County has been pretty successful. They've mentioned a couple times Get Inspired Inc. The program works by having students in classrooms maintaining aquariums with baby abalone in them, and then they outplant them. And that, I think, is responsible for a lot of the increase in green abalone in Orange County, at least. White abalone were only reintroduced since their captive breeding program in, I think, winter of 2019, 2020. And so they've been able to go back and resurvey in a limited capacity, but so far it's looking like a reasonable number of the babies that they transplanted out in these secret dive sites have been succeeding in repopulating there. So fingers crossed that keeps going because it's still a very young project. But it's they have some very cool methods with one of the concerns with introducing abalone that have been captive raised is that they're naive to predators and that they may not know how to evade predators or how to hide properly. So what they do is they transplant them in a cage where there's like walls. And the first step is to put them out with the cage walls down, and then they take the cage walls away so they still have a place to hide, but they can explore a little bit. And so they get acclimated to their surroundings in a gradual way where they're protected from predators. So fingers crossed, keep your eyes out for more news from the White Abalone Restoration Project. And that's done by NOAA Fisheries. So.
And I just sent a link to the videos of what Amy just described. Like, in the videos, they have the cage and like the rocks that they built for abalone to attach to as well.
Awesome, thank you. And I guess I'll just go ahead and chime in since we didn't have another question so far. Please feel free to put those questions in the chat. My question that I had for you is when I'm down in the tide pools, I definitely see a few abalone, not like a ton of them, but they definitely catch the attention of visitors. In terms of how, most of us that are volunteering or out in the tidal areas, how is going to be the best way to interpret the protection and the state of abalone to the average visitor?
That's a really great question. I know, speaking for our team as a whole, we are huge fans of the role that education can, of course, play in conservation. And in terms of, the protections that are for abalone right now, I talked about the Abalone Recovery and Management Plan. So they all are especially protected, and that was mostly in part due to human impacts from overfishing. And so also there's, the ecological role that abalone play. And they can be, once you start to get to know them, become a very charismatic species. I know within the past year or so, we've all kind of fallen in love with abalone. And once you really get to know them as a species, they're just like the coolest thing to find, especially still living out in the intertidals, because I know it's pretty common to find shells washed up, as you can see in my background photo here. But finding one living is pretty special as well.
Excellent, thank you. Got another question then from Scott here. What constitutes a violation when observing human behavior?
Yeah, so the data that we used for our human impacts was from MPA Watch. So there's the MPA Collaborative. So this MPA Watch program exists throughout MPAs, across the region. And so MPA violation, of course, is going to differ depending on which MPA you are in. So like Kai mentioned, the state marine reserves have the strictest regulations. There's absolutely, no matter where you are, there's no take from the intertidal zone. But so for the state marine reserve, there's also no take offshore. The state marine conservation areas do have some limited recreational fishing and whatnot. And so the violation would be dependent on what the regulations are for that specific MPA that the survey was being conducted in. So it could have included, organisms take from the tide pools, or even, take of shells from the tide pools as well. And those violations could have also accounted for maybe like offshore fishing that was seen that wasn't allowed in the specific region that they were in.
All right, Romy just popped into the chat and said life cycle question mark. So Romy, if you want to go ahead and unmute and clarify your question, that would be great.
Hi, I just want to know how would they mature into adulthood and how many years they last?
I know their lifespan definitely ranges between species. To my knowledge, some can grow to be 30, maybe 40 years old. Of course, Amy, always jump in and correct me if any of this is wrong. And we did mention that abalone are broadcast spawners, so they, release their gametes out into the water. And so they do have a larval stage before they kind of establish themselves on a substrate, which is where they will grow. And that is why organisms such as Crestos coralline algae is a very critical species for abalone recruitment and establishment because it releases some chemical cues that let those larval abalone know that like this is a good spot to come and establish themselves.
Yeah, and they actually have a really short larval stage. So they only have a few days like just swimming around before they settling down as well. And that's why they really need to be in a dense populations and in proximity so that they can fertilize sperm, eggs, and then they just like really settle down in just a few days.
Yeah, I can add a little bit too that, so since they're broadcast spawners, they're releasing their eggs and sperm. They have separate sexes that, do they change over their life, Michaela, or are they the same? Why do I have that in my head? I think that they're the same their whole life. So they will settle on the substrate, and a lot of times they're preferentially settling where there are chemical cues, where there's good sense, whether that's coralline algae or close to adult abalone that they can smell. Baby abalone will be very, very difficult to find because they take shelter in a lot of different habitats. They've been shown to hide out under the adult abalone, kind of like under their skirts of the epipodia. And then some have been found under sea urchins, nesting in sea urchin hollows. If you have too many sea urchins in an area, it can be bad for the adults, but if you have some sea urchins in an area, they can create habitat for the babies. And so they hide out just about anywhere that you might find a small limpet. They're very difficult to find. And they get to like a reproductive age at around 7 years, I think, and then can live, I think published ages are like 15 to 20 years, but I've seen ones that are much older than that. I was recently doing surveys in Northern California and found an abalone that I had seen in the same location, like exactly the same rock 12 years before. So they can stay in the same location. They have kind of a territory, and that's their home turf for most of their lives.
Awesome. Thank you for providing that background on their life cycle. And we definitely want to be conscious of everyone else's time. Thank you so much for joining us. Were there any other questions that we have before, we kind of reach our last 10 or 7 minutes or so? You can feel free to unmute as well. Go ahead and ask our speakers here or Dr. Henry if there's any additional questions that you guys have.
So this is, go ahead, Warren.
All right. Is there any plans to expand the restoration into San Diego County?
You mean with the white abalone?
Any of the abalone?
Well, I don't think that, well, I'm honestly not sure. San Diego is near the southern limits of Black Abalone Range, natural range. And so a lot of restoration has focused more in the center part of the range. Central California, the Big Sur coast, the Channel Islands have had historically much bigger populations. And so restoration has focused on maintaining those populations and kind of inching out from there. So if it does get to San Diego, it will take a little bit of time. It seems like green abalone are relatively healthy. They are found both intertidally sometimes and also in the subtidal. And so green abalone populations are probably doing pretty okay. I think that it's actually possible that there are sites where white abalone have been restored near San Diego. It's secret because they don't want to have poachers. They're very sensitive. There's only a couple locations that they've found remnant populations. I know that one of the remnant populations where they found white abalone that were used for breeding was within the San Diego area, offshore, and that I wouldn't be surprised if restoration for white abalone is ongoing there or is planned for there. I don't know about other species.
Thank you.
So my question, maybe your advisors gave you this during your presentation, but having gone through the research project, what did you learn from doing it that you might do differently another time, or what data did you not collect that you might think would be very useful, and what would you see as next steps to continue on the research that you did?
Goodness, that's a tough question because our assessment was so complicated and it did continue to build on other assessments and other protocols. And, of course, throughout, we developed this whole project management plan at the beginning of the year. And then, of course, as the year continued, a lot of things changed. At first, we were planning on collecting all of our own human impacts data. And then when we discovered this, large data set that has been existing for years that contained the same information that we were looking for, we decided that was a more robust direction to go to for, that analysis. And then I think when it comes to, deciding on the final recommendations, if we had just looked at, the positive, negative, and neutrals that we developed, we probably, if you just looked at that straight on, it probably would have given a little bit different of an answer than if we were to have taken in our own qualitative assessment and have also addressed, what can be, impacted through, restoration can, help with biotic interactions if those were negative, and, enforcement and education, as you guys are all, volunteers in the national park, that can have a big impact on human impacts as well. And so I think the thing to learn is, the nature of its complication. But I think just the development of this assessment on top of, the previous assessments that have existed, we're just learning more and more about what abalone need and, which areas can provide what they need.
One thing we didn't look at was nearshore current, and that was also what is recommended to analyze when looking at successful abalone recruitment. So I think that's something also that we would have wanted to, but with limited time, we decided to just focus on remote sensing with kelp biomass data.
Yeah, if I can chime in, because these are students who have been working on this project for about 9 months, and they have graduated, and so this is a relatively short-term project. In terms of things that I am personally interested in about black abalone and what would be fascinating to know, I know that Kai had thought about this at the beginning of the year, but whether there are larvae in the water, whether they're settling successfully is one question, but whether there is a stock of them that when they're spawning other places, if those currents, like Carmen was mentioning, do transport the larvae to places they can be. For the small handful of individuals that are found on the shore, some of the genetics of where are those ones most closely related to? Are they ones that have been there since the populations went extinct, or are they ones that have more recently colonized from larger populations in the Channel Islands? I think getting a sense of some of the dynamics of where are these natural populations coming from and what's their structure would be particularly fascinating. And I think that work that Northwest Fisheries or Southwest Fisheries Science Center is doing on captive raising them is really the next most important thing that needs to happen in order to take more steps within Orange County to restoring abalone populations there.
Sounds like they'll have their PhD programs lined up then.
There's plenty
Thank
of
you.
projects for
That's-
everybody.
Yep, thank you. That was great. It was a great presentation.
Yeah, I want to thank you all again. Thank you, Michaela, Carmen, Kai, and Dr. Henry, of course, for your time today and coming to speak with us and share what you've learned through your studies and analyses of the habitats and the populations here in Southern California. And with that, I think unless we have any last questions, I think I'll end it there and stop the recording. But yeah, we got quite a few thank yous coming in on our chat. I'm not sure if you guys are checking in on that, but very appreciative of your time and contributing for us here.
Thank all the volunteers too for the work that you're